Today we're going to look at Chapter 4 over cell structure and function. So let's start with when was most of the history of the cell established? And that was in the 17th century or 1600s. In the 1500s, we were really just using magnifying lenses to examine things like the quality of cloth. Cloth merchants would use these kind of fancier magnifying lenses to help them in their day-to-day processes. And then Hans and Zacharias Janssen, as well as Robert Hooke, improved upon those magnifying lenses by using some of the first compound microscopes. Compound microscopes have two lenses. Then we had Antoine van Leeuwenhoek in the mid-1600s. He actually started out as a cloth merchant, but he was able to use a kind of modified simple light microscope to observe living organisms in pond water. He wasn't using a compound scope, he was using kind of a fancier magnifying lens. And then in 1683, Leeuwenhoek was able to observe bacteria under his microscope. In 1883, Robert Brown discovers the nucleus in plant cells. 1838, Matthias Schleiden proposes that all plants are made up of cells, and this was the beginning of the cell theory that was later expanded upon by Theodor Schwann and Rudolf Virchow. And eventually we ended up with this idea that not only do all cells come from pre-existing cells, but cells can be considered living organisms, and some organisms are unicellular, whereas some are multicellular, and multicellular organisms generally exist as either made up of animal or plant cells. So this was essentially the cell theory, but following up this kind of boom of microscopy in the late 1800s, we had in 1931 and 1965 invention of the electron microscopes. First, the transmission electron microscope, later followed up by the scanning electron microscope. And we'll talk more about a lot of these. So first, let's talk about Robert Hooke and his amplified or improved compound microscope. You can see a picture of himself and his compound microscope on the left, and he is the scientist that first observed plant cells. He was looking at a slice of cork, and he actually noticed that the compartments looked like the compartments or the places where monks lived. And those compartments where monks lived were called cells. So he actually came up with the moniker cell for what we consider kind of a living unit. So discovering the cell wasn't all at once. It did begin or had a primitive inception about 300 years ago. We started using really kind of modified magnifying glasses before the kind of early to mid 1600s when what we would consider a real microscope was invented. So Leeuwenhoek's simple microscope was one of the first real microscopes. And the microscope, like Leeuwenhoek's and any of those that followed, including compound microscopes like that of Robert Hooke, or later the scanning and transmission electron microscopes, they have only improved this field of microscopy and helped us discover more than just the cell. So this is actually what Anton van Leeuwenhoek's microscope looked like. He was a Dutch scientist. You could see that there is a point for the specimen. There is a place to look through, or a tiny pinhole. That's where the modified magnifying glass would be located. It really only allowed for about 270 times magnification. Here's Rudolf Virchow, who contributed to the cell theory. So he was one of the scientists that kind of went against the idea of spontaneous generation, that something like fleas could arise from dust or maggots from flesh. So he went against this accepted theory of spontaneous generation and said that cells are what give rise to other cells, or pre-existing cells give rise to more living things, and that multicellular and unicellular organisms must have that cellular basis. And so in the 1800s, Rudolf Virchow was one of the key scientists adding to the concepts we consider now the cell theory. So you can see here, since the late 1800s, cell research has provided us with the three concepts collectively known as the cell theory, which we've talked about a bit, but here they are all laid out. So again, Hooke coined the term cell in 1665. We had advances in the 1830s on microscopes, and we added to this idea of what a cell is. And this eventually became known as the cell theory and includes the following. A cell is the basic unit of life. All living things composed of one or more cells. Organisms may be unicellular or multicellular. And all new cells arise from existing cells. So cells give rise to more cells. All living things are composed of at least a cell, making them unicellular or multicellular. And probably the biggest tenet is the cell is the basic unit of life, both structurally and functionally. It is allowed to do all of the complex processes that anything living can do, all by itself. So here are just a couple of different types of cells. For example, you have on the left nasal sinus cells. Then you have onion cells right in the middle. And then on the far right is Vibrio tasmaniensis, which are bacterial cells. And you can see that we have the most detail with the far right. That's because it's being viewed using a scanning electron microscope. So not only are these different organisms, but they're being viewed with slightly different microscopes. The two on the left were using light microscopes, compound light microscopes, and they cannot get as much detail as the image on the right using an electron microscope. Here are two more images. So you can see Salmonella bacteria on the left viewed with a light microscope, and then a scanning electron micrograph is on the right. See how much more detail we can get of that Salmonella bacteria using an electron microscope. So both transmission and scanning electron microscopes are going to allow for a lot more detail and a lot more magnification than a regular light microscope. So here we see a picture of a regular light microscope on the left and then a dissection scope on the right. So these essentially utilize some type of either lamp source or outside source of light in order to view a specimen. They are both compound microscopes, so we're going to use at least two sets of lenses to view our specimen. You can see on the left, the light microscope has various objective lenses that can be used or kind of turned to face the specimen, and that would modify the total magnification of your specimen. So it could increase the magnification and make the detail much better. So let's move on and talk a little bit more about cells. Well, let's look at their size first. Most cells range in size from about 1 to 100 micrometers. So that's millionths of a meter in diameter. So we can look at some things that we're more familiar with, something like the size of an apple, or a housefly, or humans, or even the tallest redwood tree. These are all things that you could see with the human eye. You would need a light microscope in order to see most eukaryotic cells, and these are some of the largest cells in the animal and plant kingdom. Then something like a prokaryotic cell that's even simpler and smaller, you would never be able to even kind of pick out with really low magnification light microscopes. And to see the fine detail with something like Salmonella bacteria, for example, we saw in an earlier slide, it would be best if you had at least a scanning, if not a transmission, electron microscope. But whether it's a eukaryotic or prokaryotic cell, these types of cells are still bigger than something like a virus or a protein or even a singular piece of DNA, and especially an atom. So they're kind of right in the middle between things you can see with the human eye and things that we're definitely going to need one of the highest types of magnifying lenses, something like an electron microscope, to see. All right, so speaking of cells as the basic structural unit of life, well, there's a way to study cells, and if you're doing that, it's called cytology. Cytology is the study of cells, but it's looking at all the functions that cells can have. These functions that match up with what living things have as characteristics, like the ability to maintain homeostasis, the ability to respond to stimuli. These things are also what individual cells have, even if they're a part of a tissue or an organ or a multicellular individual. So a characteristic of all cells is that they carry out certain life functions, and you've learned about some of those already back in Module 1. There are different unicellular organisms that can perform all of these functions by themselves. Other cells form tissues and then form organs in order to carry out an organism's life functions. So you can see that in multicellular organisms, these functions are performed in a shared manner. So they're performed by different groups of cells. And these functions include the following: ingestion, digestion, respiration, transport, homeostasis, synthesis, secretion, excretion, egestion, irritability, movement, and reproduction. So let's look at each one in a little bit more detail. Well, ingestion is the ability to take in nutrients. It includes cells when they absorb things. So they're going to bring maybe a fluid or solid particle into the cell. So ingestion is kind of a take-in process. Digestion is the breakdown or kind of compartmentalization of those products that were taken in. So digestion is the ability to break food or nutrients down into simpler forms so it can be utilized. Then we have respiration. That's the ability to release energy from the breakdown of food molecules. So here we have processes that kind of are related to each other. You must take in nutrients, digest the nutrients by breaking them down, and then utilize the energy created from those breakdown particles in powering other cellular processes. A lot of times this produces byproduct material that must be expulsed from the cell. And this is something or a process we see in a lot of multicellular organisms. We also have transport, homeostasis, and synthesis. So transport is just the ability to distribute or circulate molecules from one part of the cell to another. So cells generally can participate in transcytosis or active transport from one cell organelle to another. Homeostasis is the ability to maintain internal stability no matter what's going on outside the cell. It's also known as a type of regulation. So it's the ability of the cell, for example, to maintain water balance or the amount of certain ions that are both inside and outside their cell membrane. And then a lot of cells are going to need to be able to synthesize, to put stuff together, to take small monomers and put them together to make complex molecules, like a nucleic acid or a carbohydrate or a protein. Cell secretion usually follows a lot of digestive processes. We can get rid of any type of product we consider waste. If it's a waste product, we would specifically call that generally excretion. But if we're removing something that is non-soluble, that's undigested waste, we'll call that egestion. So it just depends whether or not that waste is soluble or non-soluble. Soluble is excretion, egestion is something it's non-soluble, but with both, we're getting rid of it. We're letting it leave the cell. The reason secretion is sometimes separate from excretion and egestion is secretion can be the release of something made by the cell. Maybe something like mucus or sweat made by glands. It's not necessarily a waste product, but you might see secretion used as a catch-all, including terms like excretion and egestion. What is meant by a cell could be irritable is it has the ability to respond to stimuli. So it has the ability to figure out where it's at. Certain unicellular organisms can also move about in their environment. And that ability to move is common more in prokaryotic than eukaryotic cells, but we see things like cilia and flagella on all types of cells. And then reproduction. If cells come from pre-existing cells, cells must have a way to reproduce. And that ability allows them to produce more cells. And I keep throwing these two terms out at you, prokaryotic and eukaryotic. These are the two basic types of cells. Prokaryotic cells do not have a true nucleus. They don't keep their DNA in a specialized compartment or space. Instead, they just have this area where they keep their genetic material called the nucleoid region. A eukaryotic cell has a true nucleus. It has this structure with a compartment that can keep its genetic material separate from other parts of the cell. All eukaryotic cells are far more complex and contain organelles, whereas we see a more simpler form of life in a prokaryotic cell. An organelle is essentially a tiny organ for a cell. Just like our organs carry out our day-to-day processes, the organelles of cells are tiny cellular structures that carry out a specific function necessary for individual eukaryotic cells to survive. So the biggest difference between prokaryotic and eukaryotic, prokaryotic doesn't have a nucleus, eukaryotic does, but we're also going to look at all of the specialized organelles in eukaryotic that really set them apart. I highly recommend this video. I'll make sure to link it and make it available to you. It's a good overview after you've looked at the rest of this PowerPoint to come back and review. All right, so more on prokaryotic and eukaryotic cells. Prokaryotic cells tend to be found in domain archaea and bacteria. They're usually a little bit older than eukaryotic cells, which are in domain eukarya. Almost all prokaryotic cells are single-celled, they're unicellular organisms, they're very simple, they have no nucleus, and they're 100 times smaller than eukaryotic cells. The larger eukaryotic cells can be single-celled, but also multicellular. Animal cells, plant cells, fungi, and protists, these are all included in domain eukarya, and all of the cells that make up these types of organisms are considered eukaryotic. And that's because they have a true nucleus. They also have increased compartmentalization thanks to their many organelles. Now, if we want to be really general and just say whether you're prokaryotic or eukaryotic, all cells contain blank, these are the things we can list. All cells have a plasma membrane. They have a boundary that gives them an inside and outside. All cells have cytoplasm, which consists of at least some organelles and a fluid to hold them in. That's the cytoplasm. They have DNA as a hereditary blueprint. Of course, eukaryotic cells put their DNA in a nucleus, and prokaryotic cells don't, but they both have genetic material. And ribosomes. Ribosomes are common to prokaryotic and eukaryotic cells, and these are going to be important for the formation or construction of proteins. But of course, there's differences between prokaryotic and eukaryotic cells. Eukaryotic cells have a nucleus, prokaryotic cells don't. That's the main difference between the two. Prokaryotic cells, instead of having a nucleus, have a nucleoid region where you'll find their genetic material. Eukaryotic cells also have membrane-bound organelles. They have these compartments with specialized functions, whereas prokaryotic cells are going to be not only smaller than eukaryotic, they tend to lack a lot of these specialized organelles. They do have ribosomes, but they don't see anything like a mitochondria or a chloroplast or a central vacuole in prokaryotic cells. So looking at prokaryotic cells, what do they have? What makes them special? Well, they do have some specialized surface features. Nearly all have at least a cell wall. That's a stiff protective coating. A lot of cells contain peptidoglycan. Bacteria would be an example. So a lot of these prokaryotic cells have a specialized protein that makes them a little bit more recognizable as a certain type of bacteria, and a lot of these tend to be more pathogenic. But not all prokaryotic cells are pathogenic or cause disease and have that stiff protective coating. They at least have a kind of modified or rigid cell wall, but this is going to be much more rigid and much more stiff than even a plant cell wall. Some are also going to be able to produce adhesive capsules, or slime layers, or even attachment pili, tiny hairs that allow them to stick to parts of their environment. Some also produce these pili for the transfer of genetic material. So this is kind of a form of genetic reproduction, but we're not actually reproducing the entire genome. And some are going to feature flagella. These flagella are used for movement. Think of them kind of as a tail or a rotor for propulsion. So here is our basic prokaryotic cell. We can see the pink pili, and that is also going to be a capsule or slime layer. Some capsules or slime layers help bacteria hide in multicellular organisms, so they're not picked up by that organism's immune system. We can also see that under the capsule or slime layer is a cell wall, followed by a plasma membrane. Plant cells, which are eukaryotic, also have a cell wall, but we don't see peptidoglycan in the cell wall of eukaryotic cells, only prokaryotic cells. And the cell wall of prokaryotic cells is much more rigid than that found in plant cells. Also, all prokaryotes have a cell wall, not all eukaryotic cells. The cytoplasm is within the plasma membrane, and you can see that we have a couple of ribosomes. We have an area where the genetic material is, the nucleoid region, and they're pointing out not only the flagellum for propulsion, but also a plasmid, which is a circular piece of DNA that in some bacteria codes for a special ability, like the ability to glow in the dark or resist antibiotics. So to continue on, prokaryotic cells have that nucleoid region. It's generally going to be a circular strand of DNA as opposed to kind of a double helix shape that's separated out and kind of scrunched into chromosomes. We might find some plasmids, but not all prokaryotic cells have plasmids, but they're common. You might find some food granules in that cytoplasm. You may or may not have some folds that function as light harvesting folds or light harvesting structures. Those are rare in prokaryotic cells, but something like cyanobacteria would have structures similar to that we see in photosynthetic organisms, like plants, where they can absorb sunlight and turn that into chemical energy for themselves. And most all of them are going to contain ribosomes. But let's focus a little bit more on eukaryotic cells. Both unicellular and multicellular organisms can have eukaryotic cells, but they all have organelles, including that true nucleus or compartment for their genetic materia. And that genetic material is going to sit within that enclosed space, and that makes them or really sets them apart from prokaryotic cells. All eukaryotic cells are a part of domain Eukarya, and they consist of plants and animal cells, protists and fungi, but we are going to focus more on plant and animal cell structure. So here's kind of the basis of plant and animal cell structure. So you can see that they do differ slightly, but they both have a nucleus. They both have organelles or compartments to carry out separate functions. One of the biggest differences is we're going to see in a plant cell, a cell wall, kind of like what we saw with the prokaryotes, just less rigid, where we only see a plasma membrane and no cell wall with the animal cell. So plant cell structure, we get a cell membrane and a cell wall. Animal cells, just a cell membrane, just that barrier. Of course, there's other differences, but we'll get into those as we move through the next couple of slides. Here's just another image of a typical animal and typical plant cell. So again, a really good slide to come back and look at or study. Very common organelles to both types of cells are things like the nucleus, the rough and smooth endoplasmic reticulum, mitochondria, different parts of the cytoskeleton like microtubules or intermediate filaments. A lot of these are going to be slightly modified, so you wouldn't necessarily see the exact type of cytoskeletal element in plants and animals, but they do have this structural organization component. You're also going to see things like the Golgi apparatus. That's very common to both types. You will not see a cell wall in animal cells. You will not see a central vacuole in animal cells. And you're unlikely to see certain types of cell junctions or even something like a chloroplast, which plants use to capture light energy and create their own chemical energy. And we're going to look at each one of these differences in more detail as we continue on. So all cells contain DNA as their genetic material. In some cells, like eukaryotic cells, it's put in a nucleus. But there's also other shared organelles and structures, a plasma membrane, that boundary, a cytoplasm that consists of a fluid to hold all those organelles, and ribosomes. These are shared commonly among, essentially, prokaryotes and eukaryotes. So let's take a closer look at that plasma membrane. What makes it a great boundary? What makes it protective but also allow for the transport of substances into and out of the cell? Well, the main characteristic of the plasma membrane is that it's semipermeable. That semipermeable membrane structure means that it gets to decide what comes in and what stays out, what the cell takes in or what it expulses, and it's made up primarily of phospholipids, which are a type of lipid molecule that have a phosphate head. That gives this phospholipid structure both a hydrophobic and hydrophilic side, meaning that parts of the phospholipids have to sequester or hide away in the median part of the cell membrane. Also embedded in this phospholipid bilayer are tons of integral proteins, or big proteins that go all the way through the membrane. Some are channels, some are carriers, so they can be communication portals, or the regulatory passageways for certain molecules and ions. So here's that basic structure. You can see all of the red heads of the phospholipid molecule, and then the lighter pink or red, those are the tails of the phospholipid molecules. And we have two layers of these phospholipid molecules with their tails, which are hydrophobic, sitting away from the inside and outside of the cell, or pointing towards the middle of the membrane. We also have these blue integral proteins embedded within the lipid bilayer, meaning at least parts of them are lipid-like and can sit very easily within this fat-like plasma membrane. Notice the heads of the phospholipids are pointed away. These are the hydrophilic parts, or the parts that like water. So these are the areas of the phospholipids that can hang out in the watery or near the watery exterior and watery interior of cells. There's also surface glycolipids that help mark cells and identify them as different types. And on the inside, there's an organizational cytoskeleton that helps support and shape cells and even allows them to kind of move about. The cytoplasm itself consists of cytosol plus organelles. The cytosol, that is the jelly-like fluid inside a cell within which the organelles are suspended. Remember, the organelles are those tiny organs or cell structures that carry out their own function. All the fluid and structures inside the plasma membrane are essentially the cytoplasm. It's essentially located between the plasma membrane and the nucleus, and we wouldn't consider anything outside the cell the cytoplasm. It's mostly water, 70 to 80%. It might contain certain ions, certain breakdown products like monomers, maybe glucose, amino acids, nucleotides, or even fatty acids and glycerol. Ions would be sodium, potassium, calcium, other elements. And a lot of the cytoplasmic streaming that occurs, which is the motion of the cytoplasm in a cell that results in coordinated movement of the cell's contents, is carried out by the cell's cytoskeleton. So again, that cytoskeleton within the cytoplasm provides a lot of organization and shape to the cell. So speaking of the cytoskeleton, let's define it. It's a network of protein fibers within the cytoplasm. It holds the cell together, gives it shape, helps it move. It's a very good structural support element for the cell. One type of organelle that creates a cytoskeletal element is the centrosome. The centrosome is actually a region that contains centrioles, and centrioles are paired organelles that create microtubules. Microtubules are one of the prominent elements of the cytoskeleton, used as molecular highways for motor proteins to move different types of organelles from one location to another within a cell. Flagella and cilia, which are things like the tail or tiny hairs on cells that can move back and forth or move an entire cell, they are also going to be cytoskeletal based. Different functions again maintain shape of the cell, function in cell division, allow cytoplasm and vesicles to move, maintain cell shape, secures certain organelles, but also enables unicellular organisms to move independently. So that's talking about the flagella and cilia. The three fiber types that make up the cytoskeleton are the microfilaments, intermediate filaments, and microtubules. Again, I already mentioned the microtubules. They're made from the paired organelles called centrioles that hang out in the centrosome region, an area near the nucleus. That's their organizing site. And microtubules are also going to be involved in division of the nucleus later on when we look at cell mitosis or cell division. So let's take a closer look at what these cytoskeletal elements look like. So microtubules are made up of tubulin, and tubulin arranges itself in a tube shape. It's made up essentially of bead subunits, and it's going to be the biggest cytoskeletal element. Intermediate filaments are intermediate in size, so they're kind of in between the other two, and they're composed of rope-like bundles of various proteins. And then we have microfilaments, which are composed of actin proteins, and they resemble twisted double strands of beads. Actin is also found in muscle and is one of the contractile proteins that allow muscles to contract. So microfilaments are mainly to allow cells to move. They're very much involved in cell motility. Something like an amoeba that can put out false feet and kind of skirt or kind of crawl along is a unicellular organism that utilizes microfilaments to do so. It's also involved in types of active transport like endocytosis and exocytosis, taking in large particles or expulsing or getting rid of them. Intermediate filaments, they're going to be attached to desmosomes, which is a type of cell junction we'll look at that allows for some stretch or give, allows for some tension without tearing cells apart. And then finally, the largest of the cytoskeletal elements, microtubules, created by centrioles, are primarily used in cell division, but also are going to be highways for motor proteins to travel along to move different organelles like mitochondria and lysosomes to different areas. So here's a general arrangement of intermediate filaments, microfilaments, and microtubules in a cell. You can see the intermediate filaments and microfilaments are much closer or hang out closer to the cell membrane, so they provide a little bit more structural support, whereas microtubules are more specifically for cell division and acting as motor highways within the cell. So that leads us away from things like the plasma membrane and the cytoplasm, and even the cytoskeletal elements. But let's look now at these compartments we've been talking about, these major organelles that work together to both modify, package, and transport substances like lipids and proteins. We got to talk about the nuclear envelope that surrounds the nucleus, lysosomes, a specialized vesicle, rough and smooth endoplasmic reticulum, and the Golgi apparatus. These are all within the plasma membrane, and so it's not technically a part of the endomembrane system, but the plasma membrane does interact with a lot of these other components. So the nucleus itself has three major parts: a nuclear envelope with pores, inside unwound DNA called chromatin, and a really dark staining region, the nucleolus, where we find rRNA or ribosomal RNA made. The nucleus itself is one of the largest organelles, generally sits in the center of a cell, and it houses the genetic material or DNA. So we can see the basic structure here. We have the nucleolus, which is responsible for making rRNA, which later goes on to help us make ribosomes. We have the unwound yarn structure. That is chromatin. So it's a mixture of DNA and proteins that is not very tightly wound. It's unwound just enough for RNA, called mRNA, to read it and get instructions. And then surrounding all of that is the double-layered nuclear envelope that does have spaces or pores. And while the DNA can't leave through the pores, other things like RNA can enter and leave. Here's a better image. It's an electron micrograph of the nucleus. So you can see it's three parts: nuclear envelope with nuclear pores. We also have the nuclear envelope studded with ribosomes. Then you can see the chromatin, kind of the unwound yarn structure, and then the dark staining nucleolus. Chromatin structure is what would be kind of right here. So it's DNA double helix wrapped around 4 histone proteins to form a nucleosome. If this supercoiled, it would create a chromosome, but cells only need chromosomes when they're about to divide. So they compact their DNA from chromatin to chromosomes when they're about to divide, because that's easier to package or sort. So around the nucleus, we're going to have a sac-like organelles. Some of the main ones and the closest to the nucleus are the endoplasmic reticulum. There's 2 types of endoplasmic reticulum, rough and smooth. Rough endoplasmic reticulum is involved in protein synthesis, and that makes sense because studded or on the rough endoplasmic reticulum are ribosomes, and that's why under a microscope it appeared rough or bumpy and was given that name. So the rough ER can synthesize proteins because of the presence of ribosomes on its surface. It can also make something like a phospholipid, so that's going to be kind of a modification of a lipid by adding a phosphate group, and that can then get added to the cell membrane. Smooth ER, or smooth endoplasmic reticulum, has no ribosomes, so it didn't appear bumpy, so it got the moniker smooth. So it is not at all involved in protein synthesis. Only rough endoplasmic reticulum is involved in protein synthesis. Instead, we see a lot of functions like detoxification, metabolism, especially the metabolization of alcohol. We see storage of different types of ions. So osteocytes or bone cells have smooth ER that will actually hold on to calcium phosphate ions. Also, lipids and steroid hormones tend to be made and modified at the smooth ER and then sent to the Golgi apparatus. The Golgi apparatus is generally called the Golgi body. It's also a sac-like organelle. It's a little bit farther away from the nucleus, so you could say it sits between the endoplasmic reticulum and the plasma membrane. It's essentially the post office for the cell. It sorts, tags, packages, and even distributes the products from the rough and smooth ER, the lipids and the proteins. It has a side that receives vesicles or cell bubbles from the endoplasmic reticulum, and a side that then releases and sends those substances on their way, sometimes to the cell membrane, sometimes out of the cell, sometimes to another organelle like a lysosome. A lysosome is essentially a cell garbage disposal. It's an organelle that contains acids, and these acids are essentially digestive enzymes that can break down large macromolecules like polysaccharides, proteins, lipids, nucleic acids. It can break them down, as well as worn out organelles, into their parts. It could also destroy a disease-causing organism like a bacteria that could be engulfed or eaten by the cell or may enter a cell. So it's going to be essentially a digesting bubble inside the cell. And we don't want the stuff inside a lysosome to get out into most of the cell, because it could actually then eat the cell itself. A lysosome is a modified vesicle, and a vesicle is a specialized cell bubble that carries out a certain function. But there's also vacuoles, which are larger bubbles, and they generally have a function like containing or storing a substance. Central vacuoles in plant cells actually store water. And vacuoles are going to be larger than vesicles. And vesicles are used to transport not only solutes, but solids. And also we have modified vesicles that become things like lysosomes or peroxisomes. So here let's point out some of those structures of the endomembrane system. We have the nucleus, then we have the sac-like rough ER with the studded pink ribosomes. Smooth ER is over here. It's more tubular than sac-like, but it's always near the rough ER. And it's closer to the nucleus than the Golgi. The Golgi, in the kind of this pink-orange, has a cis face, which receives vesicles. Then it modifies packages and then sends out on its transphase vesicles either to be secreted or maybe even to combine with the lysosome and be digested. So a lot of substances that need to be secreted by a cell will have to process or go through the Golgi, which is between the rough and endoplasmic reticulum and the cell membrane. So here's just another view of the Golgi apparatus, or the Golgi body. Here it's in green, and we can see there's a couple of different pathways for what happens to the vesicles that are leaving the transphase of the Golgi. Pathway A is a vesicle contents can be destined for exocytosis. Maybe that vesicle here contains something like mucus or neurotransmitter from a neuron. Or you could have pathway B, a vesicle membrane to be incorporated into plasma membrane. So you have a vesicle made up of phospholipids, and it's meant to fuse with the plasma membrane and allow the cell to get bigger or grow. Or maybe it's pathway C, a lysosome containing acid hydrolase enzymes is going to combine with that vesicle. So these might be substances that need to be broken down further, so it combines with a lysosome to have that process of breakdown occur. Here's a simpler view of the creation of a vesicle. This is when a cell is performing endocytosis or taking in a substance. Here it's a macrophage or a white blood cell taking in a bacterium, forming a bubble or vesicle around it, and then putting that vesicle with a lysosome, which is a modified digestive organ. Once it does that, it becomes a phagolysosome, and that bacterium can be broken down and essentially destroyed. Let's look at some other cell parts. We've talked about ribosomes, I've mentioned mitochondria, and even peroxisomes, but what are they? Well, ribosomes are small organelles. They can be attached to something, like they're attached to the nuclear membrane, they're attached to rough endoplasmic reticulum, but some are free-floating. They're responsible for protein synthesis, so anywhere you find them, like on the rough endoplasmic reticulum, that structure probably also has the function of protein synthesis. We're going to use ribosomes to make protein. An example would be the synthesis of hemoglobin, which is a specialized protein that's inside red blood cells. That protein would have to be made at a ribosome. Mitochondria are known as the powerhouse of a cell because through a series of processes, they create ATP. The main process they do is oxidative phosphorylation, which is the use of oxygen to create the most ATP during a process called cellular respiration. They tend to be oval-shaped, and they actually have their own ribosomes and DNA. Peroxisomes are like lysosomes, except they contain detoxifying enzymes. So they carry out redox or oxidation reactions that can break down substances into more neutral products. They can even detoxify many poisons. So whereas lysosomes are more digestion-based, peroxisomes are more detoxification or redox-based. Here's the basic structure of a ribosome. It has two subunits. Remember, some are attached and some are free, but no matter where they're located, all ribosomes help make proteins, or they're the site of protein synthesis. Mitochondria have two membranes, an inner and outer mitochondrial membrane. The innermost part of a mitochondria is the mitochondrial matrix, and the space between the outer and inner membranes is the intermembrane space. Different processes of cellular respiration happen throughout the mitochondria, but the end product is still going to be ATP. But the process of cellular respiration that occurs within the mitochondria does require oxygen, and without oxygen, the mitochondria is not used for creating energy, and a cellular process of ATP creation called glycolysis is all that occurs. It's interesting to note that they contain their own DNA and RNA, and it is believed that both mitochondria and chloroplasts, which we'll talk about later, originated as small prokaryotes that were engulfed or taken in by a larger eukaryote, and they formed a symbiotic mutualistic relationship with that eukaryotic cell to both absorb light energy for a chloroplast, but also to make cellular energy for the mitochondria. So let's just take a minute and take a step back. Let's review what we looked at, but also some things that we've been looking at that are only in animal cells. Those include centrioles, which make microtubules, centrosomes, which contain the centrioles, lysosomes, involved in digestion. Plants don't have these things. But animal cells don't have a chloroplast. They don't have a cell wall. They don't have specialized cell junctions called plasmodesmata, or areas that contain food or granules or dye like plastids. And they definitely don't contain a water-storing large central vacuole. So there are differences between animal and plant cells. So we're being general and introducing a lot of the organelles, but let's make sure we keep them separate. So the cell wall of plant cells is a rigid structure, not as rigid as that we see on bacterial cells, but it's still a very rigid structure that's outside the cell membrane. You see it not only on plants, but also fungal and protist cells generally have cell walls, and it's made up of cellulose, which is a kind of polymer of glucose. We also see middle lamella, or a thin film between the cell walls of adjacent plant cells, so you'll see this structure when plant cells come together to build a plant tissue. Chloroplasts are also only found in plant cells. These are the organelles that also have their own DNA and ribosomes like mitochondria, and were believed to be old prokaryotes. Whereas mitochondria are involved in the process of cellular respiration, chloroplasts are involved in the process of photosynthesis, and they actually contain chlorophyll, which is a pigment that is green that absorbs sunlight and then converts that into chemical energy. A central vacuole is a lot larger than a vesicle, and a lot of plants have central vacuoles to help regulate their tonicity, or their concentration of water compared to their outside surroundings. They can also store proteins and ions and other things inside their central vacuole, but mainly the central vacuole is going to contain water. So a lot of the large vacuoles of plant cells are filled with water. Here's a chloroplast. Like the mitochondria, it has an outer and inner membrane. However, in its inner membrane, the fluid stroma also contains stacks of thylakoids, and a stack of thylakoids is called a granum. Plastids are also double membraned, but they're a lot smaller. They're found in some prokaryotes and some protist and fungi cells, but they're generally going to be a storage area for food. So chloroplasts are technically a type of plastid because they contain a green pigment, and plastids can also be storage areas for pigment, so the storage doesn't have to be for food. And chloroplasts again are sites of photosynthesis, and they're containing chlorophyll, a light-capturing pigment, and that's actually going to utilize light energy to drive the synthesis of glucose, or a sugar, from the absorption or taking in carbon dioxide and water. Other plastids are things like leucoplasts and chromoplasts. They contain different pigments, sometimes still used in photosynthesis. Other non-pigmented plastids would store food particles, again like starches or oils. So here we're pointing to, or the image is showing you, the central vacuole. Central vacuoles mainly contain water, and they're found inside plant cells. They help regulate cell tonicity. Even in animals and plant cells, we'll have a little bit of an extracellular matrix outside the plasma membrane, and this is generally made up primarily of glycoproteins and collagen, and it helps hold the cell's structure and hold it together, or even to other cells. And we also see that some of this is going to be allowing for communication cell to cell. Here is the extracellular matrix of a cartilage cell. A cartilage cell is a connective tissue cell, and all connective tissue cells have a really elaborate extracellular matrix. So just keep in mind that this is an example of how complex the extracellular matrix for cells can be. We have a lot of fibers outside the cell performing a protective structure around this mature cartilage cell called a chondrocyte. And at the very least, you'd have a couple of these fibers helping hold the cell in place or position it with other cells of the same type to create a tissue or hold an organ together. So the extracellular matrix is outside of cells. We've talked about the cytoskeletal elements inside cells, so we must acknowledge these outside fibers as well. So the ECM is another way you could say extracellular matrix. In plants, the ECM is essentially the cell wall. It's that rigid structure that's been modified to support that cell membrane in plants, but that's missing in a lot of animal cells, so you see this more elaborate fiber-like extracellular matrix there. The extracellular matrix in plant cells might be perforated by plasmodesmata, whereas we don't see those in animal cells. They're essentially channels to connect adjacent plant cells. Here are some of the types of cell junctions we'll be looking at. You see A is plasmodesmata, so that's a cell junction or kind of channel that connects adjacent plant cells. We'll also be looking at D, which is a gap junction. A gap junction are integral proteins between adjacent animal cells, so they're essentially the animal cell equivalent to a plasmodesmata. They're both communicating junctions. Then you have B, where we're looking at desmosomes. And we also have C, where we're looking a little bit at tight junctions. So there's a little bit of each in both of those. But tight junctions are impermeable to water. They're essentially kind of like sealing up a Ziploc bag with super glue. And then desmosomes allow for some stretch or tension and essentially act like a molecular zipper. So as we look or move into cell junctions, keep in mind that not all cells are bound to other cells. Some cells are free. They're able to move around like blood cells and sperm cells, but a lot are bound together, especially cells of the same type with a similar function are bound together to form tissues. And when tissues come together, if we have multiple, we can get an organ. Most organs consist of at least 2 types of tissue, but many consist of all 4 types: epithelial, nervous, muscle, and connective. So here are the types of cell junctions we're going to look at: a tight junction, a desmosome, a gap junction, and a plasmodesmata. Gap and plasmodesmata are essentially the same thing, except gap junctions are found in animal cells and plasmodesmata only in plant cells. So a tight junction is an impermeable cell junction. It holds cells together, but in the area where the tight junction occurs, nothing can pass between the cells. It's impermeable. Desmosomes use glycoproteins as linkers, and these linker proteins act like a zipper, and they allow for a little bit of stretch and give when any of these tissues are subjected to pulling forces. Both between your skin cells and the cells that make up your heart, we would find desmosomes, and this allows for some shearing or tearing pressure not to destroy these types of tissues. Gap junctions are in animal cells, whereas plasmodesmata are in plant cells. They have a very similar function. Both are communicating junctions. Here we see the orange channel-like integral proteins creating a way for small ions and other substances to go from cell to cell. In plant cells, we have the same thing, a communicating junction that's connecting cell cytoplasms, but it's connecting adjacent plant cells, whereas gap junctions were connecting adjacent animal cells. So tight junctions use integral proteins to fuse the membranes together. Desmosomes use glycoprotein anchors, and so it's essentially linking those together kind of like a molecular zipper and allowing for some stretch or shearing pressure. It allows for some give. So that also reduces the possibility for tearing under tension or pressure. And then gap junctions and plasmodesmata are both communicating junctions between cells. Gap junctions between animal cells, plasmodesmata, those are between plant cells. And we're actually going to stop there. In the next part of this lecture series is going to look at the cell membrane. In even more detail, let's examine those phospholipids again. Let's look at the integral proteins and even the glycolipids and modified sugar molecules you find on the surface of different parts of the cell membrane.